there's an endpoint in chapter 3 that I do want to get to. Hopefully we get that far. In lab today, we're going to dissect the rat first thing, we'll do a quiz, then we'll dissect the rat. And then we'll introduce the microscope. We're a little bit behind my 10 o'clock class. So I'm going to try my hardest to catch up today. Today will be the best day to catch up. We ended Thursday talking about the properties of water. We just got into inorganic compounds. If I'm not mistaken, once we were done with water, we didn't get into salts, right? So when we think about inorganic chemistry, again, inorganic chemistry or inorganic compounds, that's going to be water, it's going to be salts, it's going to be acids, and it's going to be bases. So we talked about water already, all of its cool properties, which leaves us with the other three compounds, one being salt, one being an acid, and one being a base. And there's a bunch of different ways that you can ultimately define acids, salts, and bases. The easiest way to understand, in my opinion, an acid, a salt, or a base is to take three examples. So we know that sodium chloride is a salt, right? Sodium chloride is table salt. We know that hydrochloric acid is an acid. It's in the name. And we know that, well, we don't know maybe, but I can tell you that potassium hydroxide is a very good example of a base. And if we were to take each of these three substances, if you didn't know what HCl was, or NaCl was, or KOH was, you didn't know their names, you're going to take each of these three substances and add them into a bucket of water. Get a new blue marker, one that works. Each of these substances is a compound that's ultimately created using ionic bonding. So these are weak ionic bonds. Hydrochloric acid is a weakly ionic bonded compound, as is potassium hydroxide, as is sodium chloride, like we showed on Thursday. So if we add them to water, water is covalently bonded. What's going to happen to sodium chloride when we add it to water? We saw this on Thursday. It breaks apart, right? So it's going to break apart into sodium ions and chloride ions. Hydrochloric acid will break apart into hydrogen ions and chloride ions, and potassium hydroxide. Ultimately, water will break apart each of these three substances into their individual ions. And it's here that we're able to understand which one of these three is an acid, which one of these three is a base, and which one of these three is a salt. If you dissolve some substance in water and the concentration of hydrogen ions increases, this has to be an acid. So acids are going to increase concentration of hydrogen ions in a solution. On the contrary, if you take a substance, dissolve it in water, and it increases the concentration of hydroxyl ions, which is what OH negative is, it has to be a base. And then conversely, if you take an inorganic substance, dissolve it in water, and neither hydrogen ions or hydroxyl ions are added, and what you ultimately have is a salt. So when we think about salts, salts are ionic compounds. These are compounds that are created via ionic bonding. They contain cations that are not hydrogen or hydroxyl ions. So when they're broken apart in water, we'll see that neither hydrogen ions or hydroxyl ions are created or added to the solution. And we have a slew of different functions of salts. We're going to talk about salts more than we talk about acids or bases this semester. We're going to talk about salts in a whole host of different chapters. Specifically, though, we're going to talk about salts when we get to the muscle chapter. We're going to talk about salts when we get to the nervous system, because we use salts to allow us to create electrical currents. And we need electrical currents in the body to make a nerve fire, or we need electrical currents in the body to make a muscle contract. So we're going to talk about the movement of sodium ions, ultimately from the breaking apart of salts, in order to make muscles contract and make nerves fire. Acids, on the other hand, acids are a type of ionic substance that when added to water will break apart and increase hydrogen ions. Books oftentimes refer to hydrogen ions as proton donors. And sometimes this gets people a little turned around, and it shouldn't. Anybody remember what hydrogen's Bohr model looked like? How many protons does hydrogen have? Has one proton. So there's one proton. It doesn't have any it doesn't have any neutrons. So how many electrons must it have?
One.
One, right? So this is what the Bohr model of hydrogen looks like. For me to make hydrogen, this is just regular old run-of-the-mill hydrogen. For me to turn this into a hydrogen ion, what must happen? What has to happen for hydrogen to gain a charge? A positive charge, I should say.
It has to lose an electron.
It has to lose an electron. And if it loses an electron, what's all that's left?
The proton.
One proton, right? So a hydrogen ion and a proton are the exact same thing. So if you hear about it being an acid being referred to as a proton donator, that's what they're talking about. For hydrogen to have that positive charge, it has to be stripped of the electron, which means all that's left is a single proton. Really, the only acid, we're going to talk about a couple acids this semester, but the one that we're going to talk about the most is hydrochloric acid when we get to the digestive system, since that's the primary component of our stomach acid. A base, on the other hand, a base is going to be an ionically bonded substance that when added to water will break apart and increase a solution's concentration in hydroxyl ions, or OH negative. We're not going to talk about a bunch of bases this semester. We'll talk about a couple of them when we get to the urinary system, but that's about it. And ultimately, we use something called the acid-base pH scale to better understand how acids and bases ultimately, to better define, I should say, acids and bases. Please do not spend any quality time memorizing the pH levels of anything in this chapter, right? I want you to understand the basics of the pH scale, and then as we get into other chapters, we're going to talk about, well, this has a pH of this has a pH of that. And I just want you to be able to understand the basics of the pH scale so that when we do talk about that down the road, it makes a little more sense. Ultimately, our pH scale, it runs anywhere from 0 to 14. Obviously, the lower the number, the more what it is. The more acidic it is, therefore the higher the number, the more alkaline or basic it is. Dead center is 7. 7 is going to be considered neutral. What's the quintessential example of a neutral substance in the body? One's actually a little bit basic. It's like 7.3 to 7.45.
Water?
Water. Specifically diluted, distilled water. Distilled water is your primary example of a neutral substance. All that something with a number of, a pH of 7 means is that the number of hydrogen ions produced by acids and the number of hydroxyl ions produced by bases is equal to one another, so they cancel each other out. Acidic solutions are going to be from 6.99 and below. Basic solutions are going to have a pH from 7.01 and up. And the body likes to keep things in the middle. We like to keep ourselves somewhere in the middle, 7 and all the, numbers just around it. We don't like being in extremes. Our pH scale looks something like so. And again, don't memorize any of these. That would be a waste of your time. What I will do on the test is test you on the concept. I will give you the numbers you need to answer the questions that I want you to be able to answer for me. So when you think about, blood, urine, most of our bodily fluids, they fall somewhere in this range, and we try to keep them there. The minute they get out of those ranges, we tend to get ourselves into a bunch of trouble. The best examples of things that are really pretty acidic in our body would be gastric juice, which usually comes in at a pH of 2. But when we get to the digestive system, we're going to talk about how the stomach has some methods that help to neutralize that acid so that we don't hurt ourselves in the process. The difference between each of these numbers, for example, the difference between something with a pH of 1 and something with a pH of 2 is 10 times the acidity or 10 times the alkalinity. So if we were to have a basic scale, over here is 7. And here we have stomach acid, or hydrochloric acid, as a pH of 2. And for simplicity's sake, we'll have blood here as a pH of 7. And each of these jumps between one number is 10 times the acidity or 10 times the alkalinity. How much more acidic is hydrochloric acid than blood? Each of these would be 10 times. So it would be 10, 100, 1,000, 10,000, 100,000 times more acidic. So stomach acid is 100,000 times more acidic than blood. Does that make sense? Because 10 times 10 times 10 times 10 times 10 is 100,000. And I always ask one of those questions on the test. On the test, I will say such and such has a pH of 2, such and such has a pH of 6. How much more acidic is one substance than the other? Or how much more alkaline is one substance than the other? And of course, what's going to be the most commonly missed answer for this question, which will always be one of the other choices. A lot of people like to add it. So let's say 10, 20, 30, 40, 50. 50 times will be one of the detractor answers. So don't add them, you got to multiply. Fair enough? Before we move on to organic, is any questions on salts, acids, bases, water, the inorganics? Good. So when we talk about organic compounds, now we're talking about our larger polymer chain substances. Now we're talking about our carbohydrates, we're talking about fats, we're talking about proteins, we're talking about nucleic acids, we're talking about enzymes, we're talking about ATP, things of that nature. These are going to be our biological molecules, which ultimately we utilize for a host of different substances, everything from stored energy to kinetic energy to the proteins that we use to build the cells of our body. Nucleic acids are obviously used in our DNA and our RNA. Enzymes we use to catalyze, create reactions in the body. I'm not looking for you to ultimately become an expert. I'm going to give you a brief synopsis of each of them because it's much easier to understand these substances as we see them in action throughout some of the chapters that are going to come down the road. When we talk about organic compounds, These are compounds that we have monomers to. A monomer is going to be a single unit. Polymers are going to be a bigger unit. So for example, a polysaccharide and a monosaccharide. A monosaccharide is a single sugar. You connect a number of single sugars together, you get a polysaccharide. If we take amino acids, which is the monomer of proteins, if we link a series of amino acids to one another, we get a protein, which is a polymer. The way that we ultimately create polymers is by using a process called dehydration synthesis, which, don't let these words scare you. Break them down. What does dehydration mean? If you're dehydrated, what are you lacking? Water. So take away water, and what do? What does synthesis mean? To make. So we make something by removing water. That's what dehydration synthesis means. Real simple. If I have two monomers, let's say this is a monosaccharide and this is a monosaccharide, if I take away water, I link them together and I create a chain between the two. Dehydration synthesis. The way that we take all of the monomers in our organic compounds and we link them together is by removing water. We talked about one of the cool properties of water is we use them for reactions. Well, here's a good example of us using water in a reaction in the body. We do this all the time. On the contrary, if I want to break apart a polymer, if I want to break apart a protein into amino acids, I want to break apart a polysaccharide, a long chain sugar into simple sugars, I'm going to use a process called hydrolysis. And again, don't let anatomy words bug you. Break them down and they usually make a lot of sense. What does hydro mean? Water. What does lysis mean? You're going to hear the word lysis a lot in this class. If I lyse something, what do? I break it. So I'm breaking apart something using water. Hydro, water, lysis, break apart. I'm using water to break something apart. So as we see here, if I have that polymer, right, and I want to break it down into its individual monomers, I just add water. If I take water, I'll break it apart into monomer 1 and monomer 2. That's hydrolysis. We utilize these processes all day long. As you eat a sugar, right, which is a long chain, we want to break it down into each of its individual sugars. We're going to add water to it, and we'll break it apart via hydrolysis. On the same token, if I have individual sugars that I want to link into one giant polymer so I can store it, for example, I'm going to do so by removing water, and I'll link them all together. To introduce them real briefly, carbohydrates. When we think about carbohydrates, we're thinking of what's a more generic term for carbohydrates?
It's sugar.
Sugar. All right, carbohydrates, a simple way to think about them is sugar. These come in 3 different forms: monosaccharides, disaccharides, and polysaccharides. And we consume all 3 of these. The most important monosaccharide in the body is glucose. For the human body, it's glucose. We use glucose to feed our cells. Then our cells take glucose and turn it into ATP. Disaccharides, we consume them on the regular, fructose, for example. All right? Polysaccharides are going to be long chains of single sugars linked together. We'll see an example of that too. So when we think about a monosaccharide, we think about a simple sugar. The single best example of a monosaccharide is glucose. This is our blood sugar. This is the sugar that we ultimately put into our cells, and our cells are going to utilize to create energy, specifically in ATP. Do not memorize any of the chemistry that we see on this picture. A disaccharide is when we take 2 monosaccharides and link them together via dehydration synthesis. Sucrose, lactose, maltose, good examples. What's problematic with these is these are no longer able to get into our cells, so these are too big to get through our cells, so we'd have to break them down if we wanted to add them to our cells. we use hydrolysis to do so. So now we just have two simple sugars linked together. And a polysaccharide is going to be a long chain. The most important polysaccharide of the body is glycogen. We're going to talk about, you'll hear me mention glycogen a bunch throughout the semester. Glycogen is the stored form of glucose. I'm assuming all of you ate breakfast. If not, you should have. And when you ate breakfast, I'm sure whatever you ate had some sugar in it. So now you dump all this sugar into your body. Your body's going to put that sugar, glucose, into your bloodstream. And all that sugar is going to slowly be pushed into all your cells, right? So that your cells then can be fed, turn it into ATP. Everyone lives happily ever after. If you ate more sugar than your cells need to function, now you've got leftover sugar, right? What are we going to do with that leftover sugar?
Store it.
We're going to do two things. One is we're going to store it, but we're going to store it in one of two ways. What are the two ways we store sugar? One is certainly fat, right? You eat too many candy bars, what do you get? You get heavier, right? It is what it is, right? The other way we store sugar, though, is in the form of glycogen. What we do is we take those extra glucose molecules, we link them all together, just like we see in this picture here. All right, so we have this long chain of simple sugars, and we store this in our muscles and we store it in our liver. Because your body doesn't know when you're going to eat again. You know you're going to eat at lunch. You know you're going to have dinner tonight, but your body doesn't know that. So we're going to store some of that excess sugar as glycogen. And that way, if you decide not to eat today, well, your body can still provide sugar to your body. Does that make sense? If anything's left over after that, we turn it into fat. If there's anything left over after that, it gets into our urine, which is never a good thing. We don't want sugar in our urine. Sugars make sense? The next one is our lipids. Another name for a lipid will be what?
Fat.
Fat. So lipids and fats are the same thing, or the same term, I should say. We ultimately use lipids for a number of different things in the body. We think of fat with a bad connotation, but that's unfair. The most abundant are going to be our triglycerides, the one that we're going to talk about when we talk about fat most frequently. We're talking about triglycerides. In the next chapter, we're going to talk about phospholipids. Phospholipids make up what? That's our cell membrane. Our cell membrane is primarily made up of phospholipids. We're going to talk about steroids a handful of times, but very briefly. Some of our hormones are steroid-based. We have steroids within our structures that give them a little more, they give us a little more strength. And there's a whole host of different varieties of lipids. The two that we're going to, again, talk about with the greatest amount by far are triglycerides and phospholipids. Triglycerides, anytime I tell you here's a fatty layer of some sort, I'm talking about triglycerides. When you think about the three layers of the skin, we have the epidermis, the dermis, and the hypodermis. The hypodermis is primarily made up of triglycerides. We talk about our plasma membrane, or the membrane that surrounds all of our cells. That's where phospholipids come into play. With regards to the rest of these, we'll talk a little bit about sex hormones, obviously, when we get to the endocrine system. We're going to talk a little bit about bile salts when we get to the digestive system. We'll talk about cholesterol a little bit when we get to the digestive system as well. But the two big ones that we're going to talk about ad nauseum, of the fats at least, of the lipids, are going to be triglycerides and phospholipids. Triglycerides, otherwise known as neutral fats, these are our stored source of energy. This is a great example of potential energy in the body. As we store fat in our body, whether it's surrounding our organs and protecting our organs, or just underlaying in parts of the body where you wish you didn't have the amount of fat that you may have. This is our body's way of storing energy, because this is fat that we can break down and actually use it to power the body. They come in two varieties, unsaturated and saturated. The difference between the two is the amount of double covalent bonds between the structures. I'll show you real quick what that looks like. Saturated fats have only single covalently bonded structures, so it's not quite as strong of a bonded structure as the unsaturated fats are. And at room temperature, these are going to be solid. When we think about solid fats, you're thinking about what? What's an example of a solid fat? Butter's a great example. Lard's a good example. These are all fats that at room temperature are solid. Unsaturated fats, on the other hand, are a little stronger. They're linked a little stronger because they have at least one double covalent bond. And as at room temperature, they're going to stay liquid. So now we're thinking of our vegetable oils. We're thinking of our fatty oils, our cooking oils. They're considered a little more heart healthy. So when we think about the basic structure of a fat, of a triglyceride rather, we have 3 fatty acid chains. These are all going to be singly covalently bonded. Room temperature, they'll be solid. They're not quite as healthy for us as opposed to the unsaturated fatty acids rather, which have at least one double covalent bond. Room temperature, it'll be a liquid. I don't want you to worry about these. Phospholipids. Phospholipids are just like fatty triglycerides, with the exception that instead of having three fatty acid chains, there's only 2. And where that third one would have been, we add another group which becomes a head. When you think about a phospholipid, it has the head and it has a tail. And we're able to link these heads and tails to one another and utilize a very important property of phospholipids. The heads of our phospholipids are hydrophilic. Hydro means what?
Water.
Water, and philic means loving. So these are water-loving sections. And the tail is hydrophobic, which means what? If you're phobic of something, what are you? You're scared of it or you don't like it. So we'll just call it water-hating. Bunch of haters. So if we link phospholipids tail to tail, can water penetrate? Can water penetrate to the other side? No. It cannot penetrate to the other side because it'll never get past the hydrophobic tail. The hydrophobic tail section of a phospholipid repels water, right? So as a result, what does this create? It creates a barrier, does it not? That water can't penetrate. Water can't go from this side to here, and water can't go from this side to here. It creates a barrier. And we utilize this barrier to surround our cells, which is how our cells are able to maintain their shape. So when you take a look at a phospholipid, it's got its 2 tails, where the third tail would have been, we now have a phosphorus group, hence phospholipid, which creates a structure that kind of looks like so. That head is very it's hydrophilic, so water can come up to the head. The tail is very hydrophobic. Water can't go past the tail, it repels it. We use these to create the barriers of our cells. We're going to see, when we get to the beginning of chapter 3, we'll actually see the cell membrane, and you'll just see phospholipids throughout the whole thing. We're not going to worry about steroids. We're not going to talk about steroids, but very infrequently. Proteins. The last of the big 3. When you think about proteins, carbohydrates, and fats. Proteins are very important because we utilize proteins for a whole host of very important functions. One, this is what we use as our construction material. So when we want to build something within the body, protein is what we use. You want to build muscle, you need protein. You want to build ligaments and tendons, you need protein. They play a vital role in cell function. We're going to talk at the end of next chapter in protein synthesis. And protein synthesis' main job is to produce a protein so that the nucleus can tell the cell what to do. That's how the nucleus ultimately gives commands to the cell to allow the cell to function properly. We use them as enzymes. Enzymes are utilized to help speed up reactions in the body. Some of our hormones are protein-based. Our antibodies are protein-based. So proteins are utilized throughout the body for a whole host of different functions. And the monomer of a protein is an amino acid. So a single unit for a protein is an amino acid. If we take a number of amino acids and link them together, we get a protein chain. So here we have an example of a handful of amino acids. If we link them together, we can get ourselves a protein chain, which is what we see here. Each individual amino acid being linked together via dehydration synthesis, and we create a protein chain. Proteins, and I'm not going to ask you really any questions on what I'm about to say. At least I've never have in 10 years, so I can't imagine this semester is going to be anything different. But proteins do come in a number of different shapes, and with those different shapes comes different functions, just to show you how versatile proteins are. Fats and carbohydrates, they're very important, but they're not very versatile. We use them for a couple different key things, and that's it. Proteins, we have a wider range of things that we use proteins for. We can create little single chains known as a primary structure. This is something that the cell does on a regular basis, usually to give instructions from the nucleus to the cell on how to do what they need to do. We can also take proteins and turn them into helixes, which is what we see here. And this should be very reminiscent of what structure of the body?
The DNA.
The DNA. If you take two of those and link them together, now you have the double helix of DNA. We can create sheets, which we can use for building blocks for different structures of the body. We can come up with a globular form or a tertiary structure. Think of enzymes and things like that, have this kind of a structure. Then we could take multiple tertiary structures, link them together, and we call that a quaternary structure. Best example of this is going to be hemoglobin, which is the protein which is at the center of our red blood cells. And we use our hemoglobin as the attractant for oxygen and carbon dioxide as we're trying to move those substances around the body. Again, I don't want you to get overly lost in the concept, just to know that proteins are pretty darn versatile. This is all just saying the same thing again. The last two, one is, there's three actually. Enzymes are next. An enzyme is something that's known as a biological catalyst. What does catalyst mean?
It like hinders something.
Actually, quite the opposite.
Oh.
Not a filter. A catalyst is something that prods something to happen. So for example, if you're trying to move a horse down the racetrack, what does that jockey use as a catalyst? A whip, right? That whip catalyzes that horse down the track a little quicker, right? Gives it a little more incentive to move a little faster. Biological catalysts, these are structures that increase the rate of a chemical reaction. The body is loaded with chemical reactions. We're going to go over numerous reactions throughout the semester. A catalyst is a protein-based structure that's going to help to produce. It's going to help to produce that chemical reaction quicker, with less energy. And what's great about enzymes is they're reusable. So once we use a catalyst, we can use it again and again and again and again to speed up reactions. So a good example of an enzyme creating some reaction, the purple bean-looking structure would be our enzyme. And here we have two simple sugars, two monomers or monosaccharides that we want to link together. What we're trying to do is link these two simple sugars together. This can happen on its own, and it would take so much time to do it. What the enzyme will do, it will take these two structures, link them together, and then be reused to do the same thing again and again and again and again and again throughout the body. Using an enzyme, this process will happen quicker, utilizing less energy, therefore increasing the efficiency of the body. And we have enzymes that do all hosts of different things, whether it's create or synthesize processes like this, or there's enzymes that'll be designed to take this structure and turn it into its individual monomers. It goes both ways. So when I talk about an enzyme, what's the most common system we're going to talk about enzymes in? Digestive system. For example, we're going to talk about amylase, and I'm going to tell you that amylase breaks down starches. So what amylase does is it takes a starch and it breaks it down into its individual monomers. We're going to talk about protease, which breaks down proteins. It takes a protein, breaks it down into its individual amino acids. Nucleic acids, we'll talk about briefly. Nucleic acids, now we're talking about the main composition of our genetic material. And the two major forms, of course, are going to be our DNA and RNA. When we think about our nucleic acids, nucleic acids are made up of three key structures, one of them being our nitrogenous base. We should all have heard of adenine, guanine, cytosine, thymine, and uracil before. There's going to be a 5-carbon sugar or a pentose sugar and a phosphate group. Ultimately something like so. So here is our nitrogenous base, either the adenine, guanine, thymine, cytosine, uracil, what have you, as well as our sugar and our phosphate group. This is the basic makeup of our nucleic acid. In DNA, we take two different strands of nucleic acids and we link them together, which is what we see here. So here's a nucleic acid, here's another nucleic acid, a third, and then we link one half to another half, utilizing those key bases. And which two link together always? Adenine always links with thymine, guanine always links with cytosine. This is obviously the makeup of our DNA, which we'll talk about in greater detail at the end of chapter 3 when we talk about protein synthesis. RNA, on the other hand, or ribonucleic acid. What's the big difference between DNA and RNA structurally?
Single-stranded or double-stranded?
DNA is a double-stranded structure. RNA is a single-stranded structure. The other key difference is that DNA utilizes thymine. Thymine is replaced on RNA by uracil. And we utilize RNA during the two processes of transcription and translation, which are involved within protein synthesis. So when we want to synthesize a protein in the cell, we create RNA from DNA, and then RNA, through a series of events, which we'll talk about when we get to the end of chapter 3, allows us to produce proteins in the cell during protein synthesis. And then the last of this chapter is something called ATP. ATP stands for adenosine triphosphate. And adenosine triphosphate is a cell's version of what? Or is what a cell uses for what?
Energy.
It's its energy, right? We use ATP for energy. Anytime a cell has to create a reaction, which requires energy, ATP is what it's going to use. And to see ATP in action, there's a couple different examples. The best example here, in my opinion, is this one in the middle, which I may have given you. Nope, I didn't. So what we're looking at here is a cell's membrane. Here's that phospholipid bilayer. Inside the cell on the bottom, outside of the cell on the top. And what we see here in the middle, these little purple structures in the middle, these are proteins. These are proteins that are designed to allow structures to either enter or leave the cell. In this example, they're entering the cell. The problem is, in order for these protein channels to allow some substance to enter the cell, they have to change their shape. So maybe they're open like so. So for that substance to come in, that little protein has to change its shape. It literally has to move. For that to occur, it requires energy. ATP, adenosine triphosphate, will donate a phosphate group, turning ATP into ADP, adenosine diphosphate, and that phosphate group will link to this protein and physically force that protein to change its shape, therefore allowing some reaction to take place, allowing some change in the physiology of that cell to occur, thus allowing that cell to function properly. Does that make sense? And we're going to see this in a few different chapters. We're going to see this, we're going to talk about it in its greatest detail when we get to the muscles and we get into the nervous system, because we're going to show how a muscle contracts, which is what they're showing down here. We're going to show how a nerve fires, which requires ATP as well. And there's going to be a host of different times throughout the semester where we're talking about endocytosis and exocytosis, both of which require ATP to occur.
On the ATP, the ADP,
Mhm.
is a process of dehydration synthesis?
No. No. Well, kind of. When you're talking about, taking ATP and turning it into ADP, we're going to use hydrolysis because we're taking a polymer and breaking it up, so that's hydrolysis. And then to relink it, we'd use dehydration synthesis. Does that make sense? All right. 2 down. Which brings us to the concept of the cell. This is actually a really long chapter. This chapter will pretty much take us to the first lab exam. In lab, we're going to talk a lot about the cell and cell processes. We're going to talk a lot about tissues. And this is pretty much what we have left in lab. Now in lecture, we've still got to get through chapter 4, which is the skin, the first organ system, which doesn't really get represented that much in the lab itself. So the first part of this chapter really talks about basic biology. And when we think about basic biology, the cell is the best example of a basic biology. So these are our structural unit of all of our living cells, 50 to 100 trillion cells in the body. And when we think about the cell itself, it's composed of 3 very important structures. Structure number 1 is the nucleus. Structure number 2 is the cytoplasm. And structure number 3 is the plasma membrane. Plasma membrane is simply the outer covering of our cell. The nucleus is obviously analogous, if you will, to the brain of our cell. And the cytoplasm is everything that exists between the nucleus and the plasma membrane, which is primarily going to consist of 2 key things. One is a fluid known as cytosol. The second are a series of organ-like structures known as organelles, ribosomes, mitochondria, endoplasmic reticulums, all that kind of fun stuff. So as we look at a general animal cell, which is what we're seeing here, we can see the plasma membrane, the cytoplasm, and then obviously the colorful part is the nucleus. When we think of the nucleus is the control center of our cell. This is ultimately analogous to the brain of any cell. It contains our genetic material, all right, so our DNA, and that comes in one of 2 forms, either chromosomes or chromatin. Chromatin is relaxed DNA. Chromosomes are going to be the chromosomes that get clumped as we go into cell replication. And there's 3 very important regions: the nuclear envelope, which is the nucleus's own membrane, outer membrane; the nucleolus, which is a pit in the center of the nucleus; and chromatin, which is the area between the 2. So as we take a look at our nucleus a little closer, we can see the nucleolus here in the center. Surrounding it is chromatin, which is relaxed genetic material. And then the outer perimeter of our nucleus is a double membrane known as the nuclear envelope. The nuclear envelope we see has a series of channels known as nuclear pores. These pores allow material from the nucleus to exit out into the cytoplasm. So it makes the nucleus permeable. So that envelope, again, is a double membrane. consists of little pores that allow for things to move in and out of the nucleus, specifically out of the nucleus. And it works, the nuclear envelope works for the nucleus very similar to how the cell membrane works for the entire cell itself. It's a barrier, it's a perimeter, it's semi-permeable, things can move across it as is needed by the cell. The nucleolus, which is that center of the cell, when we look at it on the model, it's going to have a very unique color. If you look at it under stain, it's got a darker color. This is ultimately the site where we make ribosomes. Ribosomes are an organelle. And what's the function of a ribosome? Anybody remember from high school biology? We're going to see that ribosomes make proteins. So ribosomes, after produced by the nucleolus, will migrate into the cytoplasm through those little nuclear pores, and they're going to be what we use to make proteins. Last but not least is chromatin. That's the third part of our nucleus. The chromatin is relaxed DNA. It's scattered throughout this portion of the nucleus, and ultimately as the cell goes into a replication phase, when we're going to go into mitosis, this relaxed chromatin will condense into chromosomes so that we can divide that cell properly and ultimately equally distribute the DNA or genetic material into each of the two new cells. And we'll get into that in greater detail when we talk about mitosis towards the end of this chapter, towards the end of the first half of this chapter, I should say. The plasma membrane, this is the outer perimeter of our cell. It's consistent, it's basically consisting of phospholipids that create a double layer or a bilayer, allowing for those hydrophobic tails of the phospholipid to meet, creating a water-repelling layer that keeps extracellular fluid out of the cell and intracellular fluid inside the cell, creates a barrier. And as we take a look at our cell membrane, so here's our plasma membrane, primarily consisting of phospholipids, a bilayer, 2 layers, with the hydrophilic heads on the outside, the hydrophobic tails on the inside. Fluid that's outside of the cell that wants to come in is going to be repelled because these tails are hydrophobic. Fluid that's on the inside of the cell that wants to get out will be repelled because, again, those tails are hydrophobic. Having said that, though, we are able to move material in and out of the cell. There's a host of different ways that we can do so. One of the key ways to introduce the next structure is by using a series of channels, which we see within our plasma membrane. These are made out of proteins. These proteins either create channels or little carriers, if you will, that depending upon the exact makeup of them, either allow things to freely move in and out of the cell, or will move very specific substances in and out of the cells. Within the plasma membrane is a series of cholesterols. Cholesterols are used here to give the plasma membrane a little more strength and rigidity, help prevent tearing and things of that nature. On the inside of the cell, we have a series of proteins that form something called the cytoskeleton. The cytoskeleton ultimately attaches to the plasma membrane on the inside, giving our cells a very unique shape, kind of how our skeleton gives us a unique shape. And on the outside of the cell, attached to either the proteins as they protrude from the cell's membrane or attaching to the actual fatty layer itself, we have a series of sugar groups. And these sugar groups are primarily used for communication. As one cell is sitting next to another cell, these sugars allow the cells to communicate with one another. So for example, if this cell gets sick, this cell can tell a cell neighboring it that it's under distress, utilizing these little sugar groups. Or we have a cell here that's sick, and we have a series of immune cells whose job it is to detect sick cells in the body. Well, as these cells kind of patrol the area, they can realize that this cell is sick using these sugar groups, and destroy the cell. It's easier to make a new one than to allow this one to infect other ones around it. We become sicker and sicker.
What makes the sugar not get absorbed and sit on?
That's a fantastic question.
I was gonna ask you about the carbohydrates too. I mean cholesterol. How they end up getting absorbed with sugar doesn't.
Well, don't forget, cholesterol is not water soluble. That one I can answer easy. That one's not water soluble. I'll have to look up your sugar question. It's a good question. In that, I just don't know the answer. There is an answer, but what it is, I don't know. I've never actually thought of it before. So we've already talked about hydrophilic and hydrophobic, the role of the proteins, the role of the sugars. Our cells in our body are ultimately, these next 3 slides I'm going to utilize this picture to show you the difference between tight junctions, desmosomes, and gap junctions. Ultimately, each of the cells of our body, so here's cell A, cell B, and cell C. Each of these 3 cells, we have a lot of different times in our body where we link cells to one another, and we use the plasma membrane as a linking, as a way of linking cells to one another. And we do that in one of really 3 key ways. One of them, which is what we see here, is using something called the tight junction. One that we see here is called the desmosome. And the third one, which we see down here, is called the gap junction. When we think about a tight junction, imagine if you took 2 cells, you put them under a sewing machine, and you sewed them together. You sewed a seam between those 2 cell membranes. Does that make sense? Right? So you have 2 pieces of fabric, cell membrane, put them together, sew a seam. That is a tight junction. What's great about a tight junction is it prevents anything up here from getting here. It prevents stuff from moving down between the cells. Imagine this is our stomach. What's going to be up here when we eat? You put stuff in your stomach, what gets dumped into that food? Acids, right? That acid's going to be up here. Do we want that acid to get here? No. Why? If we get here, what's going to happen to this layer? It's going to burn. If we burn the underlying of our stomach, we get a what? That's an ulcer, right? One of the ways that we prevent that from occurring is by creating these little tight junctions. These tight junctions, which will ultimately seal one cell to the other, prevents material from the surface of our cells from getting down and penetrating between the cells. This creates a barrier of its own between the 2 cells. A desmosome, which is what we see here, desmosomes are part of our plasma membrane where we have little proteins protruding, and just like a zipper, those proteins kind of look like teeth. And just like a zipper, they interlock with one another. And if you zip up your zipper of your jacket, how hard is it for you to separate the 2 sides of your jacket, right? It's really difficult to do so. Well, the same thing happens here. As we create these little desmosomes, this ultimately creates an environment where now these 2 cells kind of move as one, even though they're 2 different cells. And what's important about this is it prevents the cells from being damaged as they move, or as we move those cells. And then last but not least, what we see down here is something called a gap junction. And a gap junction is a series of proteins that link to each other, and in doing so they create little channels, allowing material from this cell to get into this cell and vice versa. So we can use these as little communication channels so that one cell can communicate with the other. And in doing so, they can coordinate activities. A good example of where we're going to see gap junctions is in the heart. If this is a muscle cell in the heart, and this is a muscle cell in the heart, they're going to have little gap junctions so that one cell can tell the other cell contract, and they do it at the exact same time, which is how our heart, in part, is able to beat with a very unique rhythm. Thump, 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 thump. So again, different ways that we connect cells to one another in order to, and we use this, and this is ultimately part of the plasma membrane's function. The cytoplasm, which is our third section of the cell, the cytoplasm is ultimately the space that exists between the plasma membrane and the nucleus. So the cytoplasm is a space. Contained within the cytoplasm are two very important things. One is the cytosol. This is the fluid that exists within our cell. And the organelles, which are the mini organs, or the organs of a cell, if you will. Inclusions are things that we add to the cell. So if we bring sugar into the cell, that would be considered an inclusion. So anything that the cell brings in, it brings in to the cytoplasm. So when we think about the cytosol, again, the cytosol is a fluid that suspends within the cell, otherwise known as the intercellular fluid. The organelles are basically the organs of a cell, or the mini organs of a cell, each of which has a very unique function, just as all the organs of our body has a very unique function. And the next part of the chapter is going to go organelle by organelle by organelle by organelle to make sure that you know what all their functions are. This is that same cell picture we showed earlier, except now we've colored in all of the different organelles of the body, whether it's the Golgi or the mitochondria, the lysosome, the rough ER, smooth ER, what have you. Each of these organelles has a very unique function that allows this cell to function properly. No different than we have a very specific set of organs that have a very unique function that allow us to function properly. So, the first one, which is the one that most people have always heard of, is the mitochondria. The mitochondria is referred to as the powerhouse of the cell because mitochondria produce what? Energy. Specifically, mitochondria produce ATP. Is it the only thing that mitochondria do? No. But for our purposes, it's certainly the most important thing that mitochondria do. When we get into the muscle chapter, we're going to talk about a process called aerobic respiration. Aerobic respiration is simply when we take in sugar, we bring sugar into the cell. The cell will then break sugar down, dump it into the mitochondria, and the mitochondria will produce a ton of ATP. This ATP is what the cell is going to utilize to do all the reactions that that cell needs to do. So when we take a look at the mitochondria, we can see one up here in red, a couple of them right here as well. Mitochondria produce ATP. The next one is a ribosome. We see ribosomes anywhere where you see these little purplish dots. Some of them are existing around this organelle here, which is going to be our rough endoplasmic reticulum. We see some of them freely floating within the cytosol. These are free ribosomes. All ribosomes, regardless of their location, produce ATP. So ribosomes are the site of protein synthesis. Whether they're freely floating in the cytoplasm or whether they're part of the rough endoplasmic reticulum, all of them produce proteins. We're going to talk about the ribosomes. The only time we're really going to talk about ribosomes is when we talk about protein synthesis. After that, we're not going to talk about them a whole heck of a lot. Mitochondria we will talk about time and time again. Bringing us to the next two organelles. Both of them are considered endoplasmic reticulums. One of them is the rough endoplasmic reticulum, or the rough ER. The other is the smooth endoplasmic reticulum, or the smooth ER. The difference between the two is twofold. One, they look different. And two, their functions are different. As you take a look at this picture, this blue structure that we see here, this is the rough ER. This blue structure that you see here is the smooth ER. And as you look at them pictorially, what is the visual difference between the two? Ribosomes. The presence of ribosomes. The rough ER is dotted with ribosomes, giving it a studded appearance. If you think about like those bracelets that people wore in the 80s with the little metal studs on them, right? That same basic studded appearance is what the ribosomes give this portion of the ER. On the same token, this ER is free of ribosomes. There's no ribosomes, therefore giving it a smooth appearance, hence the name smooth ER and rough ER. With regards to their functions are very different. Now, the rough ER doesn't really synthesize proteins. All right, books talk about it synthesizing proteins. That's the job of the ribosome. The ribosome actually makes proteins. But the proteins that are made by the ribosomes will travel from the ribosome here to the rough ER here. So here's the protein as it's being produced by the ribosome. It will travel to the rough ER, and what the rough ER is going to do is two things. The first thing it's going to do is it's going to alter that protein. Basically give it a spit shot. Change it up just a little bit to make sure it's fully functional. So if you want to call that making a protein, God bless you. But to me, the guys in Detroit make the car. The people at the car wash, they just make the car look cleaner. They didn't make jack squat, right? And to me, the rough ER really doesn't produce the proteins. That's the ribosome's job. What it does do is it alters the protein, making that protein viable. It will then take this protein that's now fully functioning, put it into a vesicle, and that vesicle will be transporting that protein to other parts of the body, namely the Golgi apparatus, which is going to be the next organelle we talk about. So again, the protein is synthesized by the ribosome. It's made by the ribosome. It'll migrate into the rough ER. In the rough ER, the proteins are now going to be made functional. We're going to alter them a little bit to make them functional. We'll then package them, and we're going to transport them via these little vesicles to other parts of the body, namely, most commonly, the Golgi apparatus.
What kind of synthesis process does it go through?
One more time?
The synthesis process?
The actual synthesis process is done by the ribosome. This is and it's a process called protein synthesis. When we get to the tail end of this half of the chapter, we're going to talk about protein synthesis. And in protein synthesis, we take DNA, we produce RNA. That RNA will exit the nucleus. It'll go to the ribosome, and the ribosome will, through a process known as translation, will take that RNA molecule and turn it into protein. That protein chain that is created by the ribosome via translation of protein synthesis, that protein will enter the rough ER. The rough ER will modify it, package it, send it to the Golgi apparatus. And we're going to see here in a second what the Golgi apparatus does with it once it gets it. Does that answer your question?
Yeah, I was thinking like the dehydration and all that.
It's a different process. Now, when you're talking about linking, now here's the thing. When you're talking about how does the ribosome link amino acids to one another, now dehydration synthesis is your answer. The rough ER isn't worried about dehydration synthesis as much as the ribosome is. So yes, the linking of the amino acids to one another by the ribosome, yes, that's dehydration synthesis. Fair enough? The smooth ER, on the other hand, this lacks ribosomes, giving it a smooth appearance. Its basic function is to produce lipids, or to metabolize lipids, and detoxify the cell of things like drugs and pesticides. The smooth ER produces steroids, it produces cholesterol, and it helps to detoxify our cell. The next one is the Golgi apparatus. The Golgi apparatus, as we look at this picture, is this green structure. So here's our rough ER, which is taking proteins produced by the ribosome, altering them, putting them into vesicles, transporting them to the Golgi. Once the Golgi apparatus gets those proteins are now going to go through a series of cisterns, little folds within the Golgi apparatus, where they will then be packaged by the Golgi apparatus and taken to one of three pathways. Pathway number one is the Golgi will send these proteins out to the plasma membrane, where we will get rid of these proteins and we'll dump them out of the cell, an excretion of sorts known as exocytosis. Another concept is the proteins that are packaged by the Golgi apparatus will travel into the cell and be dissolved by lysosomes, which will help us to dissolve them. We can dump these proteins into the cell. Furthermore, our Golgi apparatus can actually communicate with the plasma membrane and add to the plasma membrane so that we can actually make our plasma membrane bigger. We can actually expand our cell size by using vesicles given to the plasma membrane and add to our plasma membrane, making our cell bigger. Ultimately, our Golgi apparatus is considered, usually most textbooks talk about it being the train tracks of the cell or the train station of the cell, because it's all about transportation. Materials that need to be transported around the cell will utilize the Golgi apparatus to do that. Primarily what we transport is proteins, but we can transport things like fats if we need to. The lysosome. A lysosome is to the cell what our stomach is to our body. The best analogy would be our stomach. In that our lysosome contains within it digestive enzymes. And anything that is brought into the cell, a lysosome will fuse with it to break it down. So if our cell was to eat a bacteria, right, which we have some cells that do that, that bacteria will enter into the cell. We will fuse that bacteria with a lysosome. Lysosome will dump its digestive enzymes. The digestive enzymes will break down whatever is brought into the cell into its manageable parts, and then the cell can do what it wants to with those manageable parts, whether it's utilize them in the cell, get rid of them via excretion, a whole host of different things. Peroxisomes. Peroxisomes are little sacs within the cell which help to detoxify the cell from harmful substances such as alcohol, formaldehyde. They break down something called free radicals. Free radicals are produced with certain metabolic processes in the body. Free radicals are dangerous because they're unstable. And the way they try to stabilize themselves is by attacking our plasma membrane. When they attack the plasma membrane, they damage the plasma membrane. And if you damage the plasma membrane too much, too often, too severely, that cell will die. Well, if every cell in your body was producing free radicals that were constantly attacking the plasma membrane, we wouldn't survive very well. Peroxisomes help to neutralize free radicals to help keep our cells healthy. Then last but not least is the cytoskeleton. Our cell has its own skeleton, and our skeleton comes in a host of different varieties, everything from microtubules, which are larger protein structures, to middle-sized fibrous substances called intermittent filaments, to microfilaments, which are smaller and thinner. We utilize our cytoskeleton for a whole host of different things, everything from giving our cell its basic structure and size. When we get into the different cells, when we get to the tissue chapter, you're going to see that every cell has a different shape. Those unique shapes are primarily provided to it by the cytoskeleton. We also use the cytoskeleton to support the organelles. Our organelles, when we take a look at, I do have a picture. When we take a look at this cell, I thought I had another one. When we take a look at this cell, those organelles aren't just freely floating within the cell. They're attached to something. And then what each of those organelles is attached to keep them fixed in place is cytoskeleton. And then last but not least, we use our cytoskeleton for appendages. Right? When you think about a flagella, what's a flagella used for?
To move.
To move the cell, right? It's a little tail-like structure on a cell that when it flaps, it moves. Well, the inside of that flagella is made up of cytoskeleton. Then this is the last of all the organelles. The last of all the organelles is the centriole. And the centriole is an organelle that's utilized in a process called mitosis. And mitosis is what? If a cell undergoes mitosis, what's it doing? It's dividing. We're taking one cell, we're turning it into two. That process is called mitosis. For mitosis to occur, one of the key ingredients is the centriole. The centriole are little rod-shaped microtubules that are found within the cytoplasm that aid in the process of mitosis. And on Thursday, what we're going to do is we're going to go over the cell model. I'll introduce you again to every single organelle. We'll go again through the functions of each of those organelles as we will label the cell model, and then we're going to utilize that material to better understand the rest of the lab that we do on Thursday. This is just a couple of different charts that kind of recap all the organelles, all their basic functions, which again, we're going to go over again in lab on Thursday. We've got a couple minutes. There are a couple of things called surface extensions. Our cells, depending on the cell, do have a series of surface extensions. And we're going to see in different cells little protrusions, little add-ons, if you will, that certain cells have, and they all have very important functions. Some cells have something called cilia. Cilia are little hair-like structures that we find on our cells. And these little hair-like fibers, it's almost like they have a head of hair, right? And the little hair-like structures, as they kind of beat in the wind, what they do is they move material that might be just on the outside of the cell. For example, in our throat. Any smokers in the house? Don't want to admit to it? I rarely get a hand up on smokers anymore. Our cells on the inside of our throat, inside of our trachea, are lined with cilia, and they beat. And what they do is they keep things out of our lungs. People who smoke, what's the one thing they tend to do a lot? They tend to cough a lot, right? Because that cilia gets destroyed by the tar in cigarettes that kind of ooze down the inside of our trachea. So where we can keep things out of our lungs by these little cilia, these little hair-like structures, people who have smoked chronically, who have lost that cilia, what happens is now that's all those same things now get into the lungs, so they constantly have to force it up, which is why they tend to hack a lung so much. It is kind of gross when you think about it. Other cells that we're going to see. So again, if you're going to take a look, if here's a cell, and it has little hair-like structures, we call that cilia. And again, these cilia, they're just little hairs, and they just kind of move, and they keep things moving that's outside of the cell. Other cells have these little finger-like projections, and these are called microvilli. And what's important about microvilli is they increase a cell's surface area. We actually are pretty small considering how much we do. And one of the ways that we can do as much as we do anatomically and still have the simple size that we have is we do a really good job of increasing surface area. We're going to talk about that a lot throughout the semester. Microvilli are one of the ways that a cell can increase its surface area so that the function of that cell is greater. And then last but not least is a flagella. Flagella is going to be a tail-like structure. Only cell in the human body that has a flagella is a sperm. So when we take a look at a sperm, this little tail-like structure is an appendage. That's a flagella, which the sperm obviously uses for locomotion, to move from the vaginal canal into the uterus, into the fallopian tube, and then into the center of the egg when we're doing fertilization. All right, that's enough for today. In lab today, did you guys bring gloves?
Yes, sir.